Assalamu alaikum everyone. So today we will discuss plaque forming unit. So what is plaque in virology? A plaque is a visible structure that forms when a virus infects a cell culture. It is a clear or translucent area where cells have been lysed, killed by the virus. Now question arises, what is plaque forming unit? A plaque forming unit is a measure used in virology to describe the number of virus particles capable of forming plaques per unit volume. What is plaque assay? A plaque assay is a method for detecting and quantifying infectious viruses in a sample. Why it's used? Plaque assays are one of the most accurate ways to quantifying infectious viruses. They can also be used to determine the effectiveness of antiviral substances. Originally, it was developed for bacteriophage virus that infect bacteria. In 1952, Renato Del Becco adapted the technique to measure animal viruses, making it is a reliable method for determining viral concentration in various types of virus. It was developed to calculate titer of bacteriophage stock. In first test tube, there is a pure virus stock. There is not any dilution. Now this viral stock is serially diluted. Now question arises, what is happening in serial dilution? Suppose in a first test tube, we take pure 10 ml virus stock. Now we will collect 0.1 ml from virus stock and transfer it into second test tube, which contains 0.9 ml water. Now the dilution of this test tube will become 10 raised minus 1, 10 ml dilution. Now for second dilution, we take 0.1 ml sample from 10 ml dilution test tube and transfer it into another test tube which contains 0.9 ml water. Now its dilution become 10 raised minus 2, 100 ml dilution. We repeat this step 5 to 6 times for more dilution. After dilution, we collect sample from different dilution and now these samples will inoculate on the petri dishes. Calculating PFU from a plaque assay, 1 in a 10 raised minus 3 dilution, there are too many plaques to count. 2 in a 10 raised minus 4 dilution, 25 plaques are observed. 3 in a 10 raised minus 5 dilution, there are no plaques because the dilution is too high. The PFU in the original sample is calculated using the formula. Formula PFU upon ml is equal to number of plaques into dilution factor upon volume of sample plated. For 25 plaques in a 10 raised minus dilution, PFU upon ml is equal to 25 into 10 raised 4 is equal to 2.5 into 10 raised 5. This tells us there are 250,000 infectious virus particles per millimeter in the undiluted sample.